Welcome back to this next video in which we are discussing multiple sclerosis. Okay, so we're currently in the process of discussing the pathogenesis of multiple sclerosis, and the point in the story that we've got to so far is that we are trying to understand how the autoreactive CD4 positive T cells and the autoreactive B cells that are directed against this protein of the central nervous system myelin are actually going to be activated, because at the moment they are in the naive state, and whilst they're in the naive state they're not actually going to be causing any problem, they're not going to lead to the disease. So how do they end up becoming activated? So we said in the previous video that it is believed that they become activated by the principle of cross-reactivity, and this is an extremely important principle to understand, because this is the way that we believe that a huge number of autoimmune diseases are actually triggered. So let me go over this principle again because it's so important. So the principle is this, that our individual who has the capacity now to get multiple sclerosis, because he has the autoreactive CD4 positive T cells and the B cells, is going to be infected by a pathogen, and we don't know in the case of multiple sclerosis what this pathogen is. However, we believe it is probably a virus. There are a huge number of candidates for what this pathogen is, and most of them are viruses. You're going to be infected by a pathogen. We don't know where this infection is actually going to occur. However, the common places that humans get infections are the respiratory tract, the gastrointestinal tract, the urinary tract, and the skin, so it's likely one of those areas. And this pathogen that you're going to get infected with is going to have an antigen that is going to contain the same peptide fragment as is present within the central nervous system myelin protein, and which is the peptide fragment that we have developed CD4 positive T cells that are directed against. So this antigen is going to be able to activate those CD4 positive T cells. In addition, this antigen is going to have the epitope that is the same as the epitope of the central nervous system myelin protein, and which is the epitope that we have autoreactive B cells directed against. So this antigen then is going to be able to activate both the CD4 positive T cells and the autoreactive B cells, and therefore is going to be able to activate that autoreactive extracellular adaptive immune response. That is the principle of cross-reactivity, that we will initiate the adaptive immune response to attack this antigen of the pathogen, but then it will cross-react with a self-protein. It's going to cross-react with the central nervous system myelin protein. So let's now actually go over how the adaptive immune response is going to be initiated in response to this pathogen. So we have talked about how you're going to initiate the inflammatory response. Remember, the inflammatory response is the innate immune response. It's extremely quick. It will occur very quickly after the pathogen has infected the tissue, whereas the adaptive immune response will take much longer, potentially days, before it's actually activated against the pathogen. So the adaptive immune response then, just taking a little bit of a step back, remember there are two major arms of the adaptive immune response. There is the intracellular arm and there is the extracellular arm. The intracellular arm involves the CD8 positive T cells, and we haven't discussed this at length because it's not believed to be that important in actually attacking the myelin protein within the central nervous system. However, know that CD8 positive T cells, it's a similar concept to CD4 positive T cells. They're all of different clones, and they are directed against a peptide fragment. The extracellular adaptive immune response, that consists of the CD4 positive T cells and the B cells working together. However, the B cells cannot be activated unless the CD4 positive T cells have been activated. So fundamentally, both of the arms of the adaptive immune response, to initiate them, you have to get activation of T cells. In the case of the extracellular one, it's the CD4 positive T cells that have to be activated. In the case of the intracellular one, it's the CD8 positive T cells that have to be activated. So you have to get T cell activation to initiate either of the arms of the adaptive immune response. Now, in order to get T-cell activation, you need a phenomenon called antigen presentation. Now, by the way, antigen can be abbreviated down to AG, and I think I will be abbreviating it down to AG from now on. So there is this very important phenomenon that we need to understand, which is the phenomenon of antigen presentation, which is the mechanism by which T cells of both types, of both the CD4 positive type and the CD8 positive type, are going to be actually activated. So you cannot activate the adaptive immune system without this phenomenon of antigen presentation. Now, before I actually describe antigen presentation to you, let me also just go back to a point that I talked about in the previous video, but which is very important to know about, although we're not going to actually discuss it in detail later on, but I want to stress this here now. When you're actually initiating an adaptive immune response against a real pathogen, like the one we have here, this pathogen will have a huge number of proteins, it will have a huge number of antigens. Each of these antigens will have a huge number of peptide fragments. You could chop up its antigens in a huge number of different ways, so you have a huge number of peptide fragments. So you can potentially activate a huge number of different clones of T cells against this pathogen. In addition, all of the proteins will have loads of different epitopes, so you can activate loads of clones of B cells if you're activating the extracellular adaptive immune response, specifically the B cells of that response. So the point is that you don't usually just get one clone of T cells activated and one clone of B cells activated. You don't just usually get one little adaptive immune response initiated. You get loads of them all being activated concurrently, loads of different clones being activated concurrently. Be aware of that, even though we're not going to actually talk about that, because all I care about as far as multiple sclerosis is concerned is that this pathogen, this antigen specifically here, is going to be able to activate our autoreactive clone of CD4 positive T cells and our autoreactive clone of B cells. So I'm only going to focus on that specific clone. I'm not going to focus on all the other clones that will be being initiated and which won't cause a problem because they don't cross-react with self-proteins. But be aware that all of those will be occurring concurrently. So, let's now begin actually discussing how antigen presentation is going to occur. So, antigen presentation is a way of activating T cells, and it is done by antigen-presenting cells, and the two major types of antigen-presenting cells are sitting right in front of us already. They are the dendritic cell and the macrophage. The dendritic cell is better at doing it than the macrophage. So these two here, these are our antigen-presenting cells. So rather than calling them sentinel cells anymore, we will now be referring to them as APCs, standing for antigen-presenting cells. Okay, remember also that inflammation will brought in loads more macrophages, so we'll have a huge number of macrophages, not just the resident macrophages anymore. So we've got loads of macrophages and loads of dendritic cells. So, macrophages and dendritic cells, they are both capable of phagocytosing pathogens, as well as neutrophils. So we've got three phagocytes here: neutrophils, macrophages, and dendritic cells. All of these can phagocytose pathogens. They can engulf them into phagosomes and then break down the pathogen within the phagosome. Neutrophils do not do antigen presentation. Macrophages and dendritic cells, when they're breaking down a pathogen, they can do something a little bit more advanced now. And that thing that's a little bit more advanced is they can chop up the proteins of that pathogen and then display the proteins or present the proteins bound to special presentation molecules that are on their surface, and they can then present them to T cells to try and activate the adaptive immune response. So, we now need to talk about the special display molecules that are on the surface of the antigen-presenting cells, which can bind peptide fragments. So just to draw a simple little picture here, on this macrophage picture that I've got here, this can represent one of these display molecules. So it's a protein on the surface of the cell, and it's got this groove in it where you can put a little peptide fragment that you're going to display on your surface, and you can take that to T cells to try and activate them. And of course, if the peptide fragment is the peptide fragment that the T cell is directed against, it may well activate. So what are the names of these display molecules? So these are called major histocompatibility complexes. So I'll write this up here. So massive word coming up here, histocompatibility So major histocompatibility complexes. Now, there are two different forms of major histocompatibility complexes, namely the major histocompatibility complex class 1 and the major histocompatibility complex class 2. And for short, these are abbreviated down to MHC class 1 and MHC class 2. Now, these two different display molecules are involved in activating the two different strands of the adaptive immune response. So this is a very, very important point to understand. So we'll spend a little bit of time here after I've selected a colour that I'd like to do this in. So we'll do it in, we'll go to aquamarine again. So, up from here, so here are antigen presenting cells. When they have phagocytosed a pathogen, they're going to break that pathogen down, they're going to break all its proteins down into little peptide fragments, and they then want to present the fragments on their surface. They can choose to present them on either MHC class 1 molecules or MHC class 2 molecules. So there are two options here, MHC class 1 or MHC class 2. Now, how does the antigen presenting cell decide which of these two major histocompatibility complexes it's going to put the peptide fragments from a certain pathogen on? Well, it depends on the pathogen and which type of adaptive immune response you think is necessary to initiate. So if you want to initiate the extracellular strand of the adaptive immune response, i.e. you want to activate the CD4 positive T cells, they need to get the peptides presented to them on MHC class 2. So, when would an antigen presenting cell decide to present them on MHC class 2? Well, it wants to do this if it has found an extracellular pathogen. So, it does this if it has found the pathogen floating around in the extracellular fluid, it phagocytoses that pathogen, breaks it down. If that's the story, then it will put the peptide fragments on MHC class 2 because it knows that that pathogen spends at least some of its time in the extracellular fluid. And I'll remind you of a very important point here. Viruses usually initiate both the intracellular and the extracellular arms of the adaptive immune response because they will spend some of their time in the extracellular fluid. They're not always inside the cells. Whereas pathogens that spend all their time in the extracellular fluid, they will just initiate the extracellular arm of the adaptive immune response. So, if an antigen-presenting cell phagocytoses a pathogen that's floating around in the extracellular fluid, it knows that that pathogen spends time in the extracellular fluid, and therefore it would be valuable to initiate an extracellular adaptive immune response against that pathogen. So when it phagocytoses it and breaks down its proteins, it will put the peptide fragments from those proteins on MHC class 2 molecules on its surface, and it can go and present those to CD4 positive T cells. So I'll put this here. MHC class 2 goes with the CD4 positive T cells. You cannot activate CD8 positive T cells from peptide fragments sat on MHC class 2 molecules on the surface of an antigen presenting cell. Okay, coming on to MHC class 1. MHC class 1 is for the intracellular adaptive immune response. So the CD8 positive T cell response. So when would an antigen presenting cell decide that it should present the peptide fragments from the proteins of the pathogen that it has just broken down on MHC class 1? Well, it would do that if it knows that the pathogen spends at least some of its time inside cells. And how does it know whether a pathogen spends some of its time inside cells? This usually happens by the antigen-presenting cell phagocytosing a portion of a dying cell. So let me explain this in a bit more detail. So if you have an intracellular pathogen, that often results in cell death. For instance, let's take the example of a virus. Viruses go inside human cells, they use the human cell to make loads of copies of themselves, and then they leave the human cell, and often they end up killing the human cell. So, when human cells die, they are phagocytosed. They break up into little bits usually, and those little fragments of the dying human cell will be phagocytosed by, for instance, macrophages and dendritic cells, which are trying to clear up the mess effectively. if they then find bits of pathogen inside of the dying bit of cell that they have phagocytosed, they can then break up the proteins of those pathogens and into little peptide fragments, and they will then put them on MHC class 1 because they know they found them inside of a human cell. So that's the way that these antigen presenting cells can know that the pathogen is an intracellular pathogen and therefore needs not just an extracellular adaptive immune response initiated to it against it to get it whilst it's in the extracellular fluid, but also an intracellular adaptive immune response. So, let me summarise what I've told you so far. So, to initiate the adaptive immune response, you have to obviously activate your T cells, and just to complete the picture up, MHC class 1 goes with CD8 positive T cells. So, of course, CD4 positive T cells and CD8 positive T cells, they are both in clones, and each clone is directed against a little peptide fragment. So we need to present peptide fragments of the proteins of pathogens to our T cells. And the way this is done is through this phenomenon of antigen presentation, which is done by antigen presenting cells. So the major two antigen presenting cells are macrophages and dendritic cells. They will phagocytose pathogens or indeed lumps of dying cell with pathogen inside of them. They'll break down the pathogen's proteins and they'll put the peptide fragments on either MHC class 1 or MHC class 2. If they found the pathogen in the extracellular fluid, they'll go on MHC class 2. If they found the pathogen inside a lump of a dying cell, it will go on MHC class 1. What is now going to happen is they are going to go and try and activate their respective T cells depending on which major histocompatibility complex they've actually put the peptide fragments on. So, what happens next then in the diagram? So we've got our antigen presenting cell that is coated with major histocompatibility complexes and peptide fragments. And I should say, of course, I'm telling you the full story here. I'm telling you about both intracellular and extracellular adaptive immune response. Of course, the response that we're really interested in is just our autoreactive response, which is only going to be an extracellular response. But of course, the pathogen that we're dealing with is potentially a virus, it is potentially an intracellular pathogen, so it will be getting intracellular responses as well as extracellular responses. It's just that none of those intracellular responses that we're going to be getting, we believe, are actually autoreactive, so they're not going to be significant to the pathogenesis of multiple sclerosis. So at some point we will stop following the intracellular story. I'm only putting this in for completion, but at some point we will stop following the intracellular story and we'll just follow the extracellular story, because that's where the autoreactive branch is, and that's the branch that we want to follow. But remember, this pathogen will be initiating loads of different adaptive immune responses, or loads of different clones. It's just that most of them aren't relevant for us to follow. So what is our antigen presenting cell going to do now? So once it's presented peptide fragments on MHC class 1 or MHC class 2, it needs to actually go to T cells to try and activate them. So where are T cells? Well, of course, we've discussed this. They're going to be within lymph nodes. So, of course, all the tissues of your body have lymphatic drainage. They have little lymph vessels that drain the tissue, and this lymph will drain into lymph nodes, which are where the T cells and B cells are sitting. So, our antigen-presenting cells, they're now going to go into the lymphatic vessels and drain to lymph nodes, and they're going to hopefully come in contact with T cells that they can activate to begin the adaptive immune response. So, let's discuss how this is going to occur then. It occurs the same for both CD4 positive T cells and CD8 positive T cells. So this will be the final bit where I discuss CD8 positive T cells, and then we'll actually stop discussing the CD8 positive T cells. So our antigen presenting cells are coming, and remember, they're going to be covered in either MHC class 1 or MHC class 2, or potentially both if they phagocytose both an extracellular pathogen and then a lump of cell with the intracellular pathogen inside of it. And the different MHC molecules that they have on their surface will have loads of different peptide fragments, so they will be presenting a huge number of different peptide fragments, and they are looking for T cells that are directed against the peptide fragment that they are presenting. Now, of course, if the peptide fragment is on MHC class 1, you can only activate CD8 positive T cells. If the peptide fragment is on MHC class 2, you can only activate CD4 positive T cells. This is the point where we're going to stop following the CD8 positive story. Yes, you will get CD8 positive T cell responses if this pathogen is indeed intracellular, but I don't care about those because none of those are going to be autoreactive. We just want to follow now the autoreactive story. We want to follow the activation of our autoreactive CD4 positive T cells and the B cells. Okay, so we're limiting our attention down now. So, let's now say we have our antigen-presenting cell that is presenting on MHC class 2 the peptide fragment of this antigen here that is the same peptide fragment that is within the CNS myelin protein. Okay, and it's trying to activate CD4 positive T cells. It might just happen to come across one of the members of that CD4 positive T cell clone that is directed against this peptide fragment, and if it does so, it will lead to the activation of that T cell. So let's draw this here. So here is our CD4 positive T cell that is autoreactive. So I'll put it's the autoimmune CD4 positive T cell directed against that peptide fragment from the CNS myelin protein that we talked about its development ages ago. Okay, so here is its picture, there's its big nucleus. So the antigen presenting cell is now going to present this peptide fragment that this CD4 positive T cell is directed against. What happens now? Well, now it's going to activate. It was previously naive, it's now going to fly into action. And what happens when a CD4 positive T cell, or indeed a CD8 positive T cell as well, what happens when they get activated, when they get their peptide that they're directed against presented to them by an antigen presenting cell, is they start dividing and dividing and dividing. So that's what's going to happen now. So I'll keep with blue for now. So the next stage is that you're going to get division occurring, and I'll just draw a little bit of this. So let's have it dividing into loads and loads of CD4 positive T cells here. So you're going to hugely expand the clone, and in fact that's a nice piece of terminology, so I'll put that here. So you get what we call clonal expansion. That clone of CD4 positive T cells that's directed against that peptide fragment, it's going to expand, the population is going to hugely go up. So the term for that is clonal expansion. Now, once we've got a huge number, and I've drawn 6 here, in reality you'll get far more than that. This cell is going to divide and divide and divide and divide inside the lymph node and produce a huge number of T cells that are all the same as it, that are all directed against the exact same peptide fragment. And what's now going to happen is these are going to begin to differentiate. So we'll change colour now. So the next thing that's going to happen is they're going to differentiate. Now, some of them are actually going to become effector CD4 positive T cells now, effector T helper cells. Others are going to become what are known as memory CD4 positive T cells. Before I discuss the ones that are going to become effector CD4 positive T cells, I just want to firstly talk about the memory CD4 positive T cells, because these are really, really important to understand in order to understand why multiple sclerosis is a disease that can go on for years and years and years and years and years and years. Because in order for it to continue going on for years and years and years, you have to have the continual maintenance of the adaptive immune response, and without the memory cells that wouldn't occur. So let me just explain this. So when they differentiate, there are 2 possibilities. They can become memory CD4 positive T cells, or they can become what we call the effector CD4 positive T cells. Now the effector CD4 positive T cells are of course the ones that are actually going to do something, but the memory ones are extremely important as well. So let's start with the memory ones. So a small fraction are going to become memory. Most of them are going to become effector CD4 positive T cells. So we might say, let's say 2 of them out of the 6 that I've drawn are going to become memory CD4 positive T cells. So the majority are going to become effector CD4 positive T cells. And remember, in reality, we're talking about much bigger numbers than 6. It's just for the basis of the picture. So these ones, what are these going to do? Well, the idea of the memory CD4 positive T cells is that they're going to replace the naive CD4 positive T cell. So remember, we activated this one here, which was a naive CD4 positive T cell, and it's divided and divided and divided and divided. If all of its progeny here became effector CD4 positive T cells, you would hit a problem. And the reason you would hit a problem is that effector CD4 positive T cells do not live very long, okay? They live for a few weeks and then they die. Remember, naive CD4 positive T cells live for years and they slowly divide to replace themselves and maintain the clone. What if you only had one CD4 positive T cell of this clone in your entire body? It's unlikely that the clone would be that small, but let's consider it as a possibility. If you only had one CD4 positive T cell of this clone directed against this specific peptide fragment, and imagine now that we're not talking about an autoimmune peptide fragment, imagine that we're dealing with an actual pathogen here. And of course we are dealing with an actual pathogen, but of course you would quite like to get rid of this clone in the case of the autoimmune disease. Imagine we're not talking about an autoimmune disease and we're just talking about a pathogen infecting you. This is a CD4 positive T cell directed against a peptide fragment of a pathogen that has actually infected us, i.e. it is a useful CD4 positive T cell clone. Forget the fact that it's going to be autoreactive, because obviously that's a whole other problem. But if we're thinking that the immune system's working perfectly and this isn't autoreactive, we don't want to lose this clone because it has actually been useful, it's been activated, and we've used it, we're going to use it to fight this pathogen that's infecting us. So the body thinks it does not want to lose the clone. If it was the case that all the progeny of the CD4 positive T cell became effector CD4 positive T cells, then they would die in a few weeks, and therefore that clone would have been wiped from your body, and that is not a good thing. In the case of this specific clone, of course, it would be a good thing because it's going to be autoreactive, but usually in the context of the activation of CD4 positive T cells, that would not be a good thing because that was an actual useful clone and we can't ever use it again is the problem. We've lost it completely because we turned all of our progeny into effector CD4 positive T cells. So that would not do at all. We need to replace the naive CD4 positive T cell to maintain the clone, and this is what memory CD4 positive T cells are for. They replace the naive one. They do exactly what the naive one does. They circulate in the bloodstream, they go into lymph nodes, they sit in lymph nodes forever, for a long, long time. They live even longer than the naive CD4 positive T cells. Some of them can live your entire lifetime. And they wait for an antigen presenting cell to come and present the peptide fragment that they're directed against, and if that happens, they will do the exact same thing as the naive one did. So it goes around in a cycle effectively. They do the exact same thing. If they get activated by an antigen presenting cell, they divide and divide and divide, and their progeny, some of them will differentiate into memory cells to replace them, and some of them will go into effector cells. So this is the way that you maintain the ability to initiate this same adaptive immune response in the future by creating memory CD4 positive T cells that will replace the naive CD4 positive T cell that you started off with. So the production of those memory CD4 positive T cells is very important for the function of the immune system. Of course, in the case of this autoreactive clone, this is the reason that this autoimmune response can be maintained for years and years and years because you're continuously producing memory ones that can be used again so that you can continually activate the adaptive immune response again and again and again. But normally, not in the context of autoimmunity, this is a very good thing because it means that if you get infected by the pathogen again, you can initiate the same clone of CD4 positive T cells that worked the previous time. So, that is memory CD4 positive T cells. Let's now go on to the effector CD4 positive T cells. So the majority of them are going to differentiate into effector CD4 positive T cells, which we more commonly call effector helper T cells. So we'll refer to them as T subscript H here. Okay, so most of the progeny of our expanded CD4 positive T cell then are going to become these effector CD4 positive T cells, and these effector CD4 positive T cells will assist in the activation of the autoreactive clone of B cells. So, let's now summarize what we have seen for the activation of our autoreactive clone of CD4 positive T cells, then we'll have a break, and in the next video we'll have a look at the activation of the autoreactive clone of B cells. Okay, so coming up here, so of course the principle of cross-reactivity was extremely important here. Here is our pathogen that's going to trigger the onset or the activation of our autoimmune response. And here is the antigen that's going to contain the peptide fragment that is also contained within the central nervous system myelin protein, and which we have an autoreactive clone of CD4 positive T cells directed against. Now, when this pathogen is in the extracellular fluid, and we've discussed the fact that it may well be a virus, but we know that viruses do spend some of their time in the extracellular fluid, when it's in the extracellular fluid, it's going to be phagocytosed by our antigen-presenting cells, our macrophages and our dendritic cells, and they will break down its proteins into little fragments. And when you've got a huge number of pathogens and a huge number of antigen-presenting cells phagocytosing them, eventually one of them will end up breaking down this antigen in the correct way that it actually ends up with that specific, really important peptide fragment. And when it does, it will put that peptide fragment on its surface bound to MHC Class 2 because it got the pathogen from the extracellular fluid, and it can now go to the lymph nodes and activate our autoreactive clone of CD4 positive T cells. So it will go to the lymph nodes, and there it might happen to meet one of the members of that autoreactive CD4 positive T cell clone that's directed against that peptide fragment. And when it does so, it will be able to activate that autoreactive clone of CD4 positive T cells. When a CD4 positive T cell activates, what happens is it divides and divides and divides to hugely expand the size of the clone, so you'll get clonal expansion here, and the cells then begin to differentiate. Some of them differentiate into memory CD4 positive T cells that replace the original naive CD4 positive T cell that was used to create the huge great population, and these can then be activated by antigen-presenting cells in the future. And they'll do exactly the same thing as the naive one did, i.e. divide and divide and divide, and some of its progeny will differentiate into memory and effector helper T cells. And the other remaining cells, the majority of the cells from the clonal expansion here, will become effector CD4 positive T cells, and these are going to be extremely important in helping B-cell activation. They will be extremely important in activating that autoreactive B-cell clone directed against an epitope of this antigen, but also against that epitope of the central nervous system myelin protein. This is the reason that helper T cells are called helper T cells. They help in the activation of B cells. So we will see the story of the activation of the autoreactive clone of B cells in the next video.